Thank you all for coming out, and thank you guys for your support. So as you know, my name is Bryan Foster. I will be presenting my dissertation research today, titled Population Dynamics of Feral Hemp, Cannabis Sativa L, across Illinois, and my committee members include, of course, Dr. Gibson, Dr. Gage, Dr. Lemme, Dr. Weber, and Dr. Lee. So to give you guys a little bit of an outline of what we will be talking about today, I will go over some introductory background information on escaped cultivar species, the specific target species of my research, which is cannabis sativa, the various varieties of cannabis, as well as its agricultural history here in Illinois. We'll get started with some introduction goals, and then we'll get into sort of the meat and potatoes of my dissertation research, where I have one population focusing on population ecology. My second chapter focuses on community and landscape ecology. Then we'll get into my weed field trials, my competition experiment, and wrap everything up with some general conclusions as well as some acknowledgements. So to start out, as we know, agriculture is a very, very important aspect to human societies, but an unintended consequence of this is that these same agricultural species can escape into the wild. And these are what we term feral plants. So again, feral plants are going to be agricultural crops that escape into naturalized communities, right? During this sort of transition into natural communities, over time, these escaped cultivar species will undergo sort of natural selection and sort of form into meatier forms of their cultivated varieties. So for example, here I have an illustration of Zea maize or corn, right? And when it escapes into the wild, it turns more weedy. So a lot of physical characteristics, a lot of physiological characteristics change when grown in agriculture compared to natural environments, right? During this time, these escaped plants can then crossbreed with sort of native relatives, right? And essentially can reduce genetic pollution in the case if you have wild varieties sort of crossbreeding with your agricultural varieties, right? We also have some other negative consequences for natural communities. So not only can you have genetic pollution of your agriculture crops, right? But these sort of escaped species can also displace native species. They can alter community structure. They can shift communities and essentially change ecosystem function, right? However, these sort of impacts can be difficult to assess at times just because of all the different complex interactions that are undergoing at once, as well as the long time frames that are usually associated with these sorts of studies. My study species is going to be Cannabis sativa L, and Cannabis sativa L encompasses a few different varieties or subspecies, right? But regardless of which subspecies or variety that we're going to be talking about, right? So these varieties are going to be herbaceous annual species in the family Cannabaceae. These were originally native to Central Asia, but through adaptive radiation, we have a ton of different physical characteristics across various varieties, right? And now this single species is grown in many different parts across the world, right? These plants are usually going to be tall. They're going to be upright, standing between 1 to 5 meters. They are going to have leaves that are palmately compound with serrated leaf edges. And this species is also dioecious, which means that there are going to be both separate males and separate females within populations, right? However, there have been some monoecious types, or individuals that have both male and female sex organs, that have been noted across different populations, right? This species is also entirely wind pollinated, so pollen from a male can travel up to 5 kilometers, and so this specific species, right, has the ability to essentially reproduce over very, very long distances. This species is also very sensitive to disturbance, right? So this specific species tends to occupy sunny, open, and well-drained soils, right? And the key characteristic for this specific species is the production of their cannabinoids. So the two main cannabinoids that we're going to be focusing on within my research are going to be THC, right, which is going to be the intoxicating chemical that cannabis produces, as well as its non-intoxicating counterpart, CBD. Right. When we talk about feral cannabis, so feral cannabis sativa is technically classified as Cannabis sativa ruderalis, right? And this just essentially refers to wild populations of cannabis. Right? So all these populations will originate from former cultivation sites where early on in their establishment, they may have characteristics or features that they had when they were grown in cultivation, but over time, as they adapt to local environmental pressures, right, some of their physical and physiological characteristics will change over time. So over time, usually these feral populations will become shorter and hardier. They'll have lower THC concentrations, and they'll be better stress resistant in terms of nutrient retention, shade, poor soils. And so here is actually a picture of a feral cannabis individual from one of my populations. Right. The other variety of cannabis that I'll be focusing on my research is what we call hemp. So hemp is essentially just the cultivated varieties of cannabis where we define an individual as hemp if they have less than 0.3% THC. And so this is the definition both Europe as well as the United States use, right? So this specific subspecies is going to be usually very tall, branchier, they're going to produce very strong stalks, they're going to have a lot of fiber, right? And so the reason that cultivation of hemp has been occurring for quite some time now is because of the utility of this plant. So various parts of the hemp plant can be used for a variety of different industries, right? So when we talk about the stalk of the plant, the fibers can be used for building materials, textiles, papers. The leaves can be used for animal bedding, and the seeds can be used for body care products, foods. And some modern applications include bioplastics, construction materials, nutrient supplements, as well as cosmetics. So this specific variety is very, very useful, very, very versatile. However, hemp in Illinois is not really a new thing, right? So during World War I and World War II, hemp was actually cultivated across Illinois, across the central and northern parts of the state, in order to create various products and textiles to be used for the war, right? And so these three older pictures here represent some of these really early cultivation sites across not only Illinois, but Kentucky as well, right? So this early production of hemp during the 1910s through the 1940s really allowed this species to sort of escape from these cultivation areas and into the natural communities. However, in 1970, hemp was essentially outlawed and deemed a controlled substance. However, we've had somewhat of a renaissance when it comes to hemp, right? With the passing of the 2014 and 2018 farm bills, where now farmers across the United States, as well as Illinois, are able to farm hemp legally, right? So now we have some more modern opportunities for this species to escape into natural communities. So this picture in the top left represents an iNaturalist, essentially iNaturalist citizen science observations of all the hemp populations that have been documented across the central and northern parts of the state, right? These populations and these observations are going to be a mix of older populations that have been around since the 40s, while others are going to be somewhat new. But overall, what we're seeing is that there are a ton of different populations scattered across the central and northern parts of the state, where historically most of the cannabis cultivation in Illinois has occurred. Right. So the overall objectives of my study for the first chapter pertaining to population ecology, I'm looking to determine which density dependent and density independent factors are contributing to the success or decay of these populations. So essentially, which non-living and living parts of the environment help regulate these populations. Within my second chapter, I focus on these same feral hemp populations. However, I want to determine how these hemp populations are affecting the native communities, right? I also want to determine environmental variables that are affecting habitat suitability as well as geographic distribution across the state. Within my third chapter, which focuses on weed field trials, I want to determine if industrial hemp has the capacity to reduce weedy species emergence. And I also want to see if including cover crops with hemp agriculture also improves hemp establishment, hemp performance, as well as suppressing weedy species emergence. And then my fourth chapter involves a competition experiment where I look at both industrial and feral cultivars of hemp. And I want to determine if weed density has any effect on hemp performance, so physical traits, yield in the form of bast fiber, as well as chemistry in the form of the cannabinoid levels. So to get into my first chapter, focusing on population ecology, right? So I essentially conducted field surveys from 2021 to 2023, where each growing season I focused on 5 different feral hemp populations. So this figure here illustrates the locations of all of my feral hemp populations within my study. During each growing season, right, monitoring occurred on a monthly basis from April to September. And during these assessments, right, for all of the individuals within the populations, I identified all individuals by stage class. So I deemed them either seedlings, immature, flowering, or senescing, right? Within these individual populations, I also randomly tagged 30 individuals. For these 30 individuals, I would assess their performance variables in the form of their heights, their number of branches, their number of leaves, as well as their longest leaves. At the end of the growing season, for the first 5 populations, I also collected biomass as well as seeds, right? So I only have seeds and biomass for my first 5 populations because that was the only year I was able to get permission from the state of Illinois to actually collect wild cannabis materials. For these same individually randomly tagged individuals, right, I also assessed their herbivory. So I looked at their leaf herbivory, right? What is the total damage per leaf? How many leaves were affected by herbivory? I looked at stem herbivory, so what percent of the overall stem was affected by herbivory, as well as flower herbivory, right? So what percent damage to flowers occurred, and what was the percent, what was the total amount of flowers affected by herbivory. For these same populations, I also collected a variety of environmental variables. So these included soil temperature, soil moisture, light availability. At the end of each growing season, I also collected soil samples for further analyses on soil pH, organic matter, phosphorus, magnesium, potassium, as well as nitrogen, right? Using GIS or geographic information systems, I was able to extract various landscape features for each population. So here I was able to get the slope, aspect, elevation, hillshade, as well as distance to farmland for each population as well. And then the final aspect of my first chapter involved a seed longevity experiment where I essentially took little nylon bags, filled each bag with 20 seeds from those populations I had seeds for. So each population had 4 total bags. And what I essentially did was I buried all these bags at one of my population sites and would go back and harvest these bags in 6-month increments up to a year. And so for these harvested bags, I would assess the germination of the hemp seeds as well as the viability of non-germinating seeds. So essentially what this experiment allowed me to do was determine how long feral hemp seeds can last in the soil seed bank. So to get into the results for this first chapter, right, I analyzed survivorship using survivorship curves, which essentially is just log transforming survivorship for populations over the growing season. So this figure here represents all of the different survivorships for each of the 15 populations. This star at top just represents the month in which individuals started flowering, right? These two bottom sort of curves here represent populations that died from unnatural causes, right? So I had one population die off from herbicide exposure. I had another population die off from mowing, right? So excluding these two, what we see is that there are diverse survivorship patterns across populations. Some populations exhibit what we call a type 1 survivorship, while other populations exhibit what we call a type 2 survivorship. So what this means is that some populations had constant mortality throughout the growing season, while other populations had most of that mortality occur at the flowering stage, right? Some other analyses I was not able to include looked at relationships between landscape variables and survivorship. So it turned out those sites that had more shade, had higher elevations, and had steeper slopes had increased mortality. So this difference that we see between survivorship of populations might actually be due to various landscape metrics, which include shade, elevation, as well as slope. I also analyzed this data, what we call an IPM analysis, which is an integral projection model. So this specific model focuses on three aspects of these populations. They focus on survival, growth, and fecundity, right? Within the survival and growth models, we ended up finding various positive and negative effects. So soil nitrogen and temperature play a very important part in both survival and growth, right? And when it comes to the fecundity side of things, so reproduction, right, we see that plant height, light availability, and soil temperature have drastically positive effects, right? So those individuals that are taller are going to produce more flowers. Those individuals at sites that have more light availability are going to produce more flowers. And those sites that have higher soil temperatures are also going to produce more flowers. So this figure here just plots hemp height versus the number of flowers, right? And we see an obvious sort of pattern going on. So the bigger you are, the more flowers you're going to produce. And so we found similar results for the survival and the growth kernels as well. So fecundity, survival, and growth are very, very size dependent, right? So the bigger you are, the higher probability of survival you're going to have, the higher probability of growth from 1 month to the next, right? And you're also going to have higher reproductive rates, right? So if you're a small hemp plant, you're probably not going to survive very long. You're probably going to stay small because the probability of growth is very low, right? And you're probably not going to produce many flowers. So these sort of aspects to these populations are going to be very, very size dependent, right? We also see a very interesting sort of relationship here between stem herbivory and flower production, right? So for those populations that had moderate levels of stem herbivory, they actually had increased fecundity. So here we sort of see a compensatory sort of trade-off within our hemp individuals, right? So when these individuals are experiencing moderate levels of herbivory, they're sort of allocating their resources to production. That might be sort of a fail-safe sort of mechanism that we see. And then some negative effects include soil moisture. So soil moisture not only negatively affected fecundity, but it also negatively affected survival as well. So again, this species does not do very well in waterlogged soils, right? So it's no surprise that we see negative effects of soil moisture on both survival as well as reproduction. Another aspect to the IPM analyses involves what we call elasticity values. So this specific analysis essentially just looks at which one of those three aspects is most important to the hemp populations. Is it survival? Is it growth? Or is it fecundity, right? And these IPM analyses also generate a value that we call lambda, right? So lambda essentially just tells us if these populations are growing, declining, or they're stabilizing over time. And so values greater than 1 indicate growth, values less than 1 indicate decline. And a value of exactly 1 indicates stability. So the lambda value I ended up getting was 1.07. So even though it is technically growing, right, this sort of indicates more of the stabilization, right? So populations are growing very, very little over time. So we could consider them essentially just stable populations, right? In terms of survival, growth, and fecundity, I distilled those elasticity values down into what we call the GFL triangle. So this is just a representation telling us which aspects are most important to our feral hemp population. So I ended up taking this triangle from a book from Franco and Silvertown, 1997, right? And so within this book, right, they essentially plot, so this range here represents sort of the range for a lot of annual species that grow and produce in one single season, right? So this is sort of the range that we would expect for annual species. And this red dot here represents our data points, or my data points for my hemp populations. So in terms of the elasticity values, it turned out that fecundity, or the reproduction of flowers, is the most important aspect, followed by surviving long enough to reproduce, then finally growth is going to be the least important aspect. And again, we sort of distill that down into this triangle, where one axis of the triangle represents fecundity, one axis represents growth, and the third axis represents stasis or longevity. And so when it comes to hemp, right, hemp is sort of right on the outside border of what we should expect annual plant species to be. But again, it's going to be fecundity that is the most important aspect to population persistence. Right? Within my second chapter, I still focus on these same exact feral hemp populations. However, this involves vegetation surveys. So during those monthly assessments, I also conducted vegetation surveys. So for each specific site, I assigned 60 individual 0.25 meter squared quadrats. Half of these quadrats were assigned to areas that did contain hemp. So these were the areas right around my randomly tagged individuals. And then the other half of those quadrats were assigned to areas that did not contain any hemp at all. And so this allowed me to easily compare and contrast the vegetation around hemp and the vegetation that didn't contain any hemp at all. Right? For all of the species that I found within these sites, I ended up collecting various functional trait data, which included height, SLA, which is specific leaf area, percent leaf nitrogen, seed mass, growth form, lifespan, pollination syndrome, seed dispersal mechanism, as well as reproductive strategy. I also collected phylogenetic data in the form of RBCL and MAC-K genes, and I collected both of these functional trait and phylogenetic data for further functional trait and phylogenetic analyses. The final aspect to this study involved a soil seed bank study. So at the beginning of each growing season, I collected even more soil samples from each population. I ended up seeding the soil samples. I took that seed soil, I placed them in little black 601 trays that were filled with vermiculite in a weed fabric. I set these trays out in a greenhouse on a heating mat that had constant irrigation. And essentially what I did was I identified all the plant species growing from these soil samples. I identified them, I tracked them, and then I would end up removing all identified species to allow for more seeds to germinate. Right? So every 4 weeks, I would take these trays, throw them in a freezer, let them stay there for 2 weeks, take them out, de-thaw them, crush them up, and essentially start that whole process over again. So the total amount of time that these trays were allowed to grow out for was about 18 weeks. I ended up analyzing the community data with what we call a Permanova, PermDisc, and NMDS ordinations with vector analyses. So NMDS ordinations are essentially a way to visualize differences across plot types, across sites, right? Essentially visualizes differences across treatments, right? So each of these ovals represents a plot type for a specific site. So remember, each site, I have two plot types. I have hemp plots and I have control plots, right? And so for each site, right, each site is going to have two ovals, and those ovals ending in H represent my hemp plots, and those ovals ending in C represent my control plots, right? The Permanova analyses determined that we did have significant differences in species communities across these two plot types, right? In the form of community dissimilarity, phylogenetic distance, and functional trait dissimilarity, right? Also ended up running some correlation analyses, which determined that hemp abundance was positively correlated with these three measures of community dissimilarity, phylogenetic distance, and functional trait dissimilarity, right? Bare ground was also going negatively correlated with all three of these measures, right? And so essentially what this means is that those areas that had high hemp abundance, right, are going to have fewer weed species competing with them, right? So another analysis that I ended up running is what we call an indicator species analysis, which just tells us which weed species are associated with which treatments or which sites, right? So the indicator species analysis determined that we had 18 indicator species for our hemp plots, but we had 53 indicator species for our control plots. So essentially what this is telling us is that feral hemp at these sites is reducing weedy species alpha diversity, right? In terms of phylogenetic distance, right, hemp plots are going to favor species from Amaranthaceae, Asteraceae, and Polygonaceae, while our control plots were more aligned with our grass species, right? So hemp is reducing alpha diversity, and grass species are essentially trying to avoid hemp as well. In terms of the functional trait dissimilarity, right, what we find is that the hemp plots are filtering for weedy species that have high values of these functional traits. So weedy species with with taller heights, bigger FSLAs, higher leaf nitrogen, and bigger seed masses were associated with these hemp plots. And so this is what this is telling us, is that these traits are sort of representative of resource acquisition strategies, right? Especially within nutrient-rich soils, right? So overall, hemp is reducing richness. They are.
I'm good. I'm actually double booked. I'm double booked, but I have two screens now, so
Hold on,
I'm trying to be
let me mute
attentive as
this
I
real
can.
quick.
Oh,
it was Dr. Antarella. All right. All right, so they're reducing species with richness, right? They're favoring species from specific families that have higher values of these functional traits, right? So some common species that were commonly found within these hemp plots included Amaranthus tuberculatus, Beluga verticulata, Ambrosia trifida, Canisa canadensis, and Persicaria virginiana. So we're seeing that hemp is actually significantly influencing these various natural communities. In terms of the soil seed bank, I analyzed this data with a similar approach using NMDS and Permanova, right? And so essentially what this told me is that we have significant difference in seed bank composition across all sites, right? I ended up calculating various diversity metrics too. So I calculated overall richness, Shannon diversity, Simpson diversity, and I plot those here in this box plot. So this box plot represents these diversity metrics for the above ground communities, and as well as the below ground and the above ground communities. So essentially what this is telling us is that the soil seed bank has higher diversity compared to their above ground counterparts. So essentially the soil seed bank is acting as a diversity reservoir, which expresses various species that are not in the above ground communities. So again, in terms of the soil seed bank, differences across sites, and these soil seed banks are much more diverse than the above ground communities. I also went ahead and did some further GIS analyses in the form of max ent models. So these models essentially predict the distribution of a species across a given area using various environmental variables as well as co-occurrence points. And so here I ended up running two different models, one that focuses on our current climate. So this specific max ent model here incorporates current climatic variables, current terrain variables, as well as current soil variables as well, right? And then I ended up creating a future model to see how this sort of distribution is expected to change over time using a middle of the road scenario for 2026 and 2024, right? So within this current max ent model, right, we see that these blue colors represent low suitability, while all of our greens, yellows, and reds represent higher suitability areas. These black points here represent the individual presence points I used to create the model. And what we're seeing is that Southern Illinois is not as suitable compared to the central and northern part of the state, right? Another aspect to these models are going to be response curves, right? And what these response curves do is they tell us why it's this area that is most suitable. And it turns out that hemp is most suitable within central and northern Illinois because these areas have moderate rainfall, they have lower elevations, they have more gentle slopes, they have intermediate clay content. So clay content helps the soils retain water, right? And hemp doesn't really like that. And these areas also have high organic matter, especially compared to the southern part of the state where we have more clay, less organic matter, we have more rainfall, we have higher elevations, and we have more steeper slopes, right? I ended up taking this current model and essentially comparing it with my future model, which produced a difference map. So this figure here on the right represents the expected shifts in suitability from our current climactic scenario with our future one. So all of the yellow coloring is the parts of the state that will not change in suitability, right? So we still see that Southern Illinois doesn't really change much in terms of suitability. So regardless of climatic shifts, we still should expect Southern Illinois to be less suitable compared to the rest of the state, right? These red areas indicate an increase in suitability, and these green areas indicate decreases in suitability, right? So these increases in suitability actually might be driven by warming temperatures in cooler counties, right? And these decreases in suitability might be due to anticipated heat or drought stress. But the overall pattern that we're seeing is that suitability within the future becomes a little bit more patchy, right? And then in terms of sort of these shifts, right, we expect the majority of the state, so about 45%, to stay unchanged, right? 31% of the state is going to increase in suitability, and 24% is going to decrease. The third chapter of my dissertation focuses on industrial hemp and weed field trials. So within this chapter, I conducted two main experiments. The first one being a seeding rate by fertility treatment, which was conducted from 2020 to 2021, right? And this invoked two different sites, right? So we had a northern site at Illinois State University. We had a southern site here at SIU. And then my second experiment involved a hemp cultivar by cover crop experiment, which lasted from 2021, 2022 down here at the Touch of Nature Center at SIU. So within the seeding rate and fertility experiments, we had two different treatments. The first one was a hemp seeding rate. So these levels included a 22, a 44, a 67, and an 89 kilogram per hectare seeding rate. Within the fertility treatment, we had a control, so no fertilizers added to these plots. We had a low nitrogen, a high nitrogen, a high nitrogen plus potassium, a high nitrogen plus high potassium plus humic acid, and then a humic acid only treatment, right? So this first figure here labeled A just represents the plot design for this specific experiment, which was a randomized block design. Within my second experiment, again, I had two treatments. The levels within the cultivar treatment included a fiber variety, which we call Jinmal, a oilseed variety, which we call field breastfeed, and a no hemp sort of control, right? Within the levels of the cover crop, we only had two levels, right? We either did not have a cover crop at all, or we did, and it was in the form of a mix of sea-cow cereal, which is rye, and trifolium repens, which is white clover, right? And so this second figure here illustrates the plot designs for this experiment, which was a split plot design. And so after the initial planting of hemp, I went out and assessed vegetation 2, 4, and 8 weeks post planting, right? Then at the end of each growing season, right, within these plots, I harvested hemp. I determined the height of the hemp, the biomass, the stand counts, and I also harvested the weedy fresh weight. Using these data, right, I analyzed them with a very similar approach in the Permanova and the NMDS ordinations. So for the Illinois State and SIU sites, right, what we end up seeing are drastic differences. So one thing to remember is that the soils at Illinois State or a northern site are more suitable for hemp. So the soils at ISU are going to have less clay and more organic matter compared to southern Illinois soils. So where we grow hemp in soils that are optimal for hemp growth, right? What we see is that both seeding rate and fertility had a significant effect on weedy biomass across both years of 2020 and 2021. Various hemp performance variables had negative effects with weedy species alpha diversity. So the taller our hemp was, the more hemp we had, the more biomass for hemp we had, the less weedy alpha diversity we ended up having. The big thing here is that we found significant effects of hemp seeding rates on community dissimilarity, phylogenetic distance, and functional trait dissimilarity. So these three figures here are from our 2020 data. And what we need to be focusing on here are these big green ovals. So these big green ovals represent the highest hemp seeding rate. And so what the vectors within these ordinations are telling us is that the highest hemp seeding rate led to the highest hemp stand count and the lowest weedy species alpha diversity. So in terms of community dissimilarity, the more hemp we have, the less weeds we have, and the more hemp stand count we have as well. In terms of phylogenetic distance, right, our highest hemp seeding rate results in less density from various plant species families, including Convolvulaceae, Portulaceae, Solanaceae, Poaceae. And in terms of functional trait dissimilarity, these really dense hemp plots are filtering for weedy species that have low SLAs, low percent leaf nitrogen, smaller heights, and smaller seed masses. So where hemp is growing and establishing very well at our northern site, it's the hemp itself, which is changing the community, right? Reducing alpha diversity. It's reducing densities of weed species from various families, right? And it's determining which traits these weed species have in terms of SLA, height, seed mass, and percent leaf nitrogen. So at our southern site, where hemp establishment was much lower, right, only the fertility treatments had any significant effect on weed biomass across both years. In terms of hemp performance, we only had one variable, which was visual score, which is just a way to visually assess the vigor and health of our hemp plants, right? So only visual score had any significant relationship with weed biomass, and that was in 2021. In terms of the effects on the weed communities themselves, right? Fertility had a significant effect on weedy community dissimilarity across both years, but only in 2020 was there an effect on functional trait dissimilarity. So these two figures on the far right represent the community dissimilarity in 2020 and 2021. And what this is showing is when we have poor hemp establishments, it's going to be the fertility treatments that are really shaping and affecting these weedy communities. And so what we need to be focusing on is this orange oval at the bottom and these red and black ovals at the top. So this orange oval on both figures represent our high fertility treatments, while the red and the black ovals represent our control and our low input fertility treatments. So essentially, we're getting more weedy species alpha diversity, right, when we don't have a whole lot of nutrients in the soil, right? So when we add nutrients into the soil, right, that really it really allows for the growth of very fast growing competitive species, right? So we'll get the highest diversity when nutrients are low. So again, where hemp establishment is not so great, it's going to be the fertility treatment really shaping the communities. In terms of the hemp cultivar and cover crop experiment, I analyzed this all with similar statistical approaches. We had cover crops have a significant effect on weed diversity in 2021. We did have hemp cultivar have a significant effect on species biomass in 2022. But the interesting result here is that our Bielbrzeski variety ended up leading to more weed species even compared to the no hemp control, right? So again, at our southern site, again, we're having very low hemp establishment, right? And because of this, we only found one hemp variable to negatively affect any aspect of these weedy species, and that was weedy species biomass in 2021. When it comes to effects on the weed communities, it was the cover crop treatment that was shaping these weedy communities. So these ovals here represent our cover crop treatment, and these ovals here represent our no cover crop treatment. So rather than the hemp itself really shifting communities, it's the cover crops. So in terms of community dissimilarity, right, the cover crops led to greater hemp abundance, greater hemp stem counts, right, greater hemp heights, while also reducing weedy species biomass and weedy species alpha diversity. In terms of phylogenetic distance, right, our cover crops are improve the densities of Amaranthaceae, but all other representatives from all other families are reduced, right? In terms of functional traits, we see that when we have cover crops, it's reducing the functional traits of height, SLA, and percent leaf nitrogen, right? So those weedy species that are going to be competing in these cover crop treatments are going to have lower heights, smaller SLAs, lower leaf nitrogens. They're going to mainly be from the family Amaranthaceae, right? So cover crops are not only improving hemp performance, but they're also reducing weedy species emergence, right? Another sort of thing to note here is that we did see cultivar effects. So our Gin Ma variety outperformed our Bielbrzeski variety by a significant margin. Within my competition experiment, this was conducted here at the Tree Improvement Center in Carbondale, Illinois. Here I had essentially two trials or two factors. I looked at hemp cultivar and I looked at weed species densities. So the hemp cultivars I included were the same Gin Ma and Bielbrzeski varieties in the weed field trials. And I also included seeds from my feral population 2 and feral population 3. The weed species I included were those that I observed to naturally occur quite a bit in our field studies, and that was Amaranthus, Tuberculatus, Setaria fabarii, Glycine max, and Lamium purpureum. The competition density treatments included a control. So all of these pots were planted with one hemp individual, right? So our control plots only included our one hemp individual. Our low competition plots had one of each weed species planted in a circular form around our hemp plant. So 4 total weed individuals. Our medium competition treatment included 3 individuals from each weedy species, so 12 total individuals. And our high competition treatment included 5 individuals from each species, so 20 total individuals. Again, another important note is that when transplanting occurred, both the hemp and the weed species themselves were all at the seedling stage. And so once transplanted, all of these individuals within all the pots were allowed to grow for a total of 10 weeks. Every 2 weeks, I took some repeated measures, which was hemp height, number of leaves, as well as leaf length, right? And at the end of that 10-week period, I took some final assessments in the form of stem diameter, biomass. I assessed their gender. I looked at fiber yield by essentially retting, drying, and removing that bast fiber and weighing it. And then I analyzed the cannabinoids with HPLC analyses. And so what I ended up finding, so I analyzed all this with two-way and COVAs, right? And so what we see is we have hemp cultivar effects, competition effects, interaction effects between both cultivar and competition, and gender effects, right? So just like our weed field trials, right, we see that GenMaw. So GenMaw is represented by these purple bars here. And again, letters that are different across each bars represent significant difference, right? So we see that our hemp cultivar is outperforming all others in terms of performance. So biomass, stem diameter, 5%, leaf length. In terms of competition, which is this low figure here. So high competition, medium, low in our control, right? We see that high weed competition is also reducing performance, right? We did see some interaction effects between cultivar and competition, especially with leaf length. So some varieties, especially Bill Bresky in our feral sessions, right, were more negatively impacted by competition compared to our GenMaw fiber variety. We did see some gender effects. So females consistently outperformed males across all cultivars and across all competition treatments, right? So females had, females were taller, they had longer leaves, they had more leaves, they had bigger stem diameters, and they had more biomass, right? In terms of chemistry, we didn't find any significant effects for competition in THC and CBD, which indicates that these cannabinoids might not necessarily be influenced by competition, right? So some general conclusions from each chapter, right? So for my first one on population ecology and hemp, it's going to rely on annual recruitment, where survival and fecundity are going to be very, very highly size dependent. We did find some density independent and density dependent factors that influence population stability. So specifically herbivory affected these populations. Soil nutrients significantly affected these populations. And we see sort of a compensatory trade-off between stem herbivory, which ended up boosting flower production. Within the community and landscape ecology chapter, right, we see that hemp is reducing native species richness, while phylogenetic functional traits sort of converge for fast-growing competitive species, right? In terms of the soil seed bank, we see that the soil seed banks are much more diverse compared to the above ground communities. And then in terms of distribution, we see that a lot of these hemp populations are going to be distributed across the central and northern part of the state, where it's the soil and climate's really driving this distribution. In terms of the weed field trials, we see that in soils that are good for hemp, right? Industrial hemp effectively is suppressing weedy species. In terms of cover crops, we see that cover crops are not only enhancing hemp performance, but also suppressing weedy emergence. And we see key differences between cultivars, right? So our GenMaw variety was consistently doing better between our Bill Bresky. In the competition experiment, right, we see that weed competition is going to reduce hemp performance. We see cultivar differences, right? So GenMaw shows superior performance and as well as resilience, right? So this suggests that industrial hemp varieties may be more immune to competition, even compared to our feral varieties. Cannabinoids were unaffected by competition, which suggests that cannabinoid levels are primarily dictated by genetics. And then again, we have very drastic differences between cultivar as well as gender, right? And so there are quite a few people I would like to thank, right? And these include my wonderful committee. So doctors Gibson, Gage, Weber, Lemme, and Lee. Gibson, I'd like to thank the Gibson lab past and present, which includes Doctors Barfnick, Thapa, Xian Lu, Jaren, master's student Shamim Ahmed for help with field work as well as data analysis. I'd like to thank some additional faculty, which include Doctors Newbig and Doctors Mary Kinzel for help with phylogenetic analyses as well as spectrometry. I'd like to thank Dr. Wim Phippen and Joe Groover from Western Illinois University for site access. I'd like to thank individual landowners, Ken Kraft, James Garlish, Mike Grubb, Joyce, Jeff, and Missy Hochstatter for even more site access. I'd like to thank Dr. Nicholas Heller and Becca Dwyer for providing additional weed species data to use from our weed field trials. I'd like to thank the Illinois Department of Natural Resources as well as the British Ecological Society for funding and permitting. Then of course, I'd like to thank my closest friends and family for their love and support throughout this journey. And so with that, I would be happy to field any questions.